Hello, everyone. I'm Shresh, and I'll be talking today about discovering genetic medicines with the AI drug discovery platform at Deep Genomics. Before I begin, I would like to acknowledge and thank all of my amazing colleagues who have made all of this work possible and are just a pleasure to work with every day. So let's begin with some statistics. Around 65% of people over their lifetime will have a disease with a genetic basis. And if we look at the number of births every year with serious genetic disorders, the number is around 8 million. And the total lifetime healthcare cost in the US healthcare system for, for a family who is dealing with a birth with a genetic disorder is around 5 million. So, in addition to the enormous emotional burden borne by people who go through this, these numbers emphasize the weight of the problem that we're trying to solve, which is to address genetic disease and find effective therapies in a short amount of time for them. So we believe that good drugs are based on a strong understanding of the underlying biology, and biology is too complex for humans to understand. AI can enable us to do a much better job of this and can massively accelerate this entire process. So our focus is on developing systems that can accurately predict molecular and cell biology, and this enables us to use our technology at every step of the drug development process, including target discovery, identifying a lead compound, which is drug discovery, preclinical research, and clinical research. We focus on mechanisms which are known to be causal, so which have been established in the literature and have been studied to cause disease. And all of the predictions made by our systems are for mechanisms that can be verified experimentally. So our AI systems help us to know which experiments to do, to know which compounds to test, and thus enable us to rapidly accelerate the process of discovery without having to compromise any of the scientific robustness. And in this presentation today, I'll talk about a story of how we used one of the predictors in our AI workbench for a mechanism called RNA splicing to discover a novel target and therapeutic candidate. And this is the first ever AI discovered therapeutic candidate that would not have been discovered without the AI system. So discovering this compound without the AI would have been impossible. So a bit of information about how the genome works. This picture is part of the central dogma of molecular biology, which describes the process by which genes are expressed from the template, which is your DNA. So your DNA is comprised of a sequence of letters, A, C, G, and T, as shown in this picture here. Your DNA gets transcribed into RNA, which is processed in several ways, including RNA splicing, which is illustrated in this figure here. So your DNA first gets converted into pre-messenger RNA, which contains both these gray chunks and the green and yellow chunks. Some parts of the pre-messenger RNA are spliced out. These are known as introns, and certain parts remain in the final mature mRNA, and these parts are known as the exons. So even though the introns are actually removed and are not coded into the functional protein, they do contain crucial regulatory logic which direct the process of splicing and changes in the intron can affect how the splicing machinery works, which is shown in this example here. So if you have a mutation in either the intron or the exon, the splicing machinery can be disrupted in a way such that, for example, in this case, the middle exon gets skipped. So the final mature mRNA contains the first exon, the third one, but not the middle one. And this leads to a truncated protein, which is non-functional. So finding these mutations is important, but also very tricky because many introns are several tens of thousands of base pairs long, and most changes in your genome do not cause any change. So the base rate for a mutation causing a splicing change or any change is very low. And experimentally verified data for that maps these mutations to their splicing impact is very limited. So even though you could train some shallow models on databases of mutations which map, let's say, a change to its splicing impact, you could not train a model which understands the core biology. There just isn't enough mutation data to do that. But can we use machine learning in a different way, which models the core biology and enables us to build systems that have an understanding of splicing in a way that you can use even small variant data sets, mutation data sets, to and use transfer learning to transfer the knowledge of these models to these data sets and fine tune them, even though they're very small. And it turns out that we can. And here I'm describing one approach to modeling RNA splicing, which we've found to be very accurate and successful. So this process consists of encoding your pre-messenger RNA, the sequence of your pre-messenger RNA as a 4-channel input, which is just the 4 characters, so A, C, G, and T. And we feed this into a neural network, and the outputs of the neural networks basically uh, designate whether each position is an intron in or an exon. So the gray portions in the pre-messenger RNA correspond to a 0 in the output, and the colored chunks, the exons, they correspond to a 1 in the output. And so in addition to the intron and exon track, we also have these special colors, the green and red ones. So the beginning of an exon is called an acceptor site. So we have that in a separate channel. We specially designate the beginnings of exons and also the ends of exons, which are known as the donor sites. So this would be the labels corresponding to the canonical transcript, but each portion of pre-mRNA can be spliced in many different ways through the process of alternative splicing. And the junctions that result from this are also added to the acceptor and donor tracks. So for example, if instead of the middle exon being spliced in as shown here, you could have a shorter or longer version of it, uh, which just changes the splicing boundary. And those other possible outcomes are also encoded into the acceptor and donor track. So our final output is enriched by an additional 5 million junctions mined from a collection of RNA-seq datasets. We run a lot of processing steps to ensure these junctions are of high quality. And this is done to give our model a full picture of what are the possible outcomes, what are the possible splicing outcomes for this portion of pre-messenger RNA, and to give it the opportunity to learn all of the possible ways in which splicing regulation can go based on the same sequence. Uh, and it turns out that by building these networks, which are fairly large and can model a large amount of context. So the network that I described in the previous slide, it uses a dilated convolution. It uses dilated convolutions to expand the context at each position in the output to around 16,000 base pairs. So every position can look into 16,000 base pairs uh, of context to inform its predictions. And in this example here, which is taken from real data, uh, you have three exons and introns in between them, and this is drawn to scale. So the introns are significantly larger than exons. And we run this through the model, which and this portion, this example was not part of the training set for the model. We see that the model very accurately identifies which portions are exonic and intronic. And it also identifies correctly the locations of the acceptor and donor site. So the acceptor site is marked as green, and the donor site is marked as donor sites for each of the exons are marked as red. And if we introduce a mutation similar to the one we had in the example slide before, and we know this mutation to cause exon splicing. So this is a mutation which has been reported in clinical databases to cause exon skipping. If we change that one position, we see that the output tracks of the model show that the exon strength has been reduced dramatically. So the model does not think that the middle portion is likely to be an exon. So it's not likely to be included with this change of sequence introduced by the mutation. And the other exons we see are not impacted. So the model predicts that after this mutation is introduced, we would have splicing in a way that the middle exon is excluded, which we can look into the functional impact of that. So if skipping this exon causes a frame shift, or it causes a stop codon to be introduced, that would lead to a non-functional protein. And so even though our model was never shown any mutations while training, the understanding that it develops for how sequence affects splicing allows us to predict the impact of these mutations very accurately without having to show it any mutation data at all. And because our models are designed in a way that they incorporate very long-range context, you can predict the impacts of mutations which are thousands of base pairs away from the exon. So, for example, certain classes of mutations known as pseudo-exon creation events are deep inside the intron. They're several thousands of base pairs away from the exon. But even in those cases, our model can very accurately predict the impact of those mutations. So let's look at how well this model performs on the task that it's trained on. So it's trained on predicting splicing junctions, locations as either the acceptor or donor sites, or classifying locations as exonic or intronic. So on the test set for this model, which is held out during training, we can predict these sites with extremely high precision. So with a false positive rate of 5%, we can identify splice junctions with a sensitivity of 91%. And this is significantly higher than classical approaches such as max and scan, which look at a very short window of about 20 base pairs around the splice junction to identify it as a splice site. And our model also performs favorably over other neural network approaches such as Splice AI, which try to address the same problem. And on different classes of mutations, the few that we looked at, which, for example, exon skipping, and other classes such as pseudo-exon activation, in which you have a portion in the middle of an intron being included, which becomes a pseudo-exon, or alternative splicing events in which you have exon extension or truncation. In all categories of these mutations, our models are very accurate at predicting the impact of them, even though these models have never seen these mutations or never seen these sequences really while training. And at 5% false positive rates on most classes of variants, except for the most challenging ones, we can predict the impact or predict which mutations are pathogenic with over 90% sensitivity. So far, we've talked about a system of that is very useful to discover which genetic mutations alter splicing. So if you look at a large database of unannotated mutations which are clinically relevant, we can identify which ones of those alter splicing, and we can look at which of these mechanisms, which of these five that I've shown here, how the mutation affects splicing. Does it cause exon skipping? Does it cause alternative splicing? And that allows us to direct functional experiments to verify the effect of the mutation. But identifying the mutation only gets us so far. Can we actually use machine learning in a way that we can find compounds that remediate the effect of the mutation? And it turns out that we can do that as well. So the modality of drug that we use for this is called a steric blocking oligonucleotide, which is a short sequence of modified RNA, which is about 20 base pairs long. And we can use our system to design this sequence of 20 base pairs in any way that we like. And you can usually these SBOs, as we call them, are designed to be complementary to a location of RNA. So they're designed to bind to a specific location of your RNA and sterically block regulatory elements to have a desired impact. So let's look at an example. So we looked at the example of this intronic mutation, which caused this middle exon in green to be skipped, which led to a truncated protein, which was non-functional. But if you have a steric blocking oligonucleotide, which is designed to be complementary to this location, you can have the reverse effect of the exon skipping. So you can design it in a way that the exon is no longer skipped and you have functional restoration of the protein. So most of these, most of the SBOs, if you design it around an exon, they would not have the desired impact. So as we saw in some of the previous examples, the introns can be quite large. So the search space of where you place an SBO to have the desired impact, let's say you want to revert exon skipping, is fairly large. And even if we've correctly identified the mutation, which is classically very difficult to do, but like our neural networks allow us to identify the effects of these mutations. Even after we've gone that far, the space of possible oligonucleotides being large means that you would have to do a lot of experiments to identify which compound has the desired effect. But you can use the predictors that we talked about so far, the predictors for splicing, and bootstrap the knowledge in the features trained by the model, and just enrich it with some of the features specific to SBOs. And it turns out that if you use a small amount of data, around 500 experimental data points, which we generated in our own lab, and you fine tune a network. In this example, we used a gradient boosted machine, and it used the features trained from our splicing predictors, and it incorporated certain thermodynamic properties of the steric blocking oligonucleotide. It incorporated things such as the number of mismatches with its target location, what is the relative location of the SBO with reference to other elements such as the end of an exon or the beginning of an exon. And by incorporating these hand designed features and plus the features from the deep learning model, we can get a very high correlation with these experimental measurements. So in the context of molecular biology, a correlation of 0.81 with an experimental value of an experimental value with something that is purely generated in silico is extremely high. And what this means is that you can vastly reduce the amount of experiments that you do if you're really confident in the predictions made by the system. You can vastly reduce the amount of SBOs you need to screen, greatly reduce the search space, and accelerate the process of finding these compounds by about an order of magnitude. So this leads to the story around DG12P1. How do we use all of the advances that we discussed, the advanced splicing predictors, which predict the impact of mutations, the oligo on and off target predictor, which allows us to reduce the amount of compounds that we need to screen and identify leads quicker. All of that led us to the discovery of DG12P1, which is the compound that we identified as a lead compound for treatment of Wilson's disease. So how does DG12P1 work? So DG12P1 is a drug candidate for the treatment of patients with Wilson's disease who harbor a specific genetic mutation, which is known as the MET645ARG mutation. So this designation of the mutation just mentions what is the sequence content of the mutation and what is the location of the mutation. So Wilson's disease impairs the body's ability to remove copper. It affects 1 in every 30,000 people worldwide. And if left untreated, it can cause life-threatening organ damage. So our AI system scanned over 2,400 diseases and over 100,000 pathogenic mutations while searching for good drug development opportunities. And by analyzing all of these data points, we were able to predict and confirm the precise disease-causing mechanism of the mutation, and thereby identify a clear therapeutic target. So if, as we see in this figure here, the purple compound, which is DG12P1, enters the cell. In healthy individuals, you have a T at a certain location in your RNA, but in patients with Wilson's disease, you have a G in the same location, which causes exon 6 in the ATP7B gene to be skipped. So to the left, your final mature mRNA without DG12P1 does not contain exon 6. Thus, these transcripts get degraded, and there's no ATP7B expression. But in transcripts with DG12P1 bound to them, exon skipping is blocked, and ATP7B expression is restored. So you have functional restoration, and this is the mechanism of action which was predicted by our AI system and was confirmed using experiments, as we'll talk about in the next slide. So this is the development pipeline for DG12P1, our lead candidate for the treatment of Wilson's disease. There were several key predictions made by our AI systems that enabled and expedited the process of discovery. And so the first thing to notice is in the first box that we've outlined here, for about 20 years, people knew that this mutation, MET645ARG, was pathogenic, but the mechanism by which it causes the disease was unknown. And so people were aware of this mutation, but they did not know how exactly it affects the cell. Thus, they could not come up with an intervention to target it. So in Q1 of 2018, this mutation showed up in a list of mutations predicted by our system to cause a change in splicing. We also predicted that the actual change in the amino acid sequence was not the result of pathogenicity. We predicted that the change in amino acid sequence does not have enough of a functional impact. So we were fairly confident that this mutation operates by causing exon 6 skipping. So this was in Q1 of 2018. Through Q2 of 2018 and Q1 of 2019, we validated the splicing mechanism. So we confirmed that indeed this mutation does cause ATP, does cause exon 6 to be skipped. Through Q2 of 2019, we screened several compounds, several SBOs to uh, identify which one does indeed restore ATB, does restore exon 6 inclusion. So if you look at the timeline, from the candidate being from the mutation being nominated by our AI system to indeed finding 12 lead compounds which actually address the mutation, that took only about 1 year. And beyond that, we did several experiments to confirm the tolerability and efficacy of the compound. And in Q1 of 2020, we actually declared DG12P1 to be our lead candidate, which would go on to further investigations as a investigatory new drug. So the timeline from discovery to development here is much shorter to what is the industry standard. So we brought the development timeline to about 2 years, whereas the industry standard without using machine learning would be about 5 to 7 years. And this was enabled by the use of our predictive systems, by the use of our splicing predictors in this case. And we were confident that this compound would work in advance, and thus we were able to invest our energy and resources in it. And finally, I would like to look into beyond this one specific mutation that we talked about. We've created a dashboard to show the universe of splicing mutations and show how vast this problem is and what is the addressable space that can be addressed by our technology. So I'm going to switch to the demo now. So in this dashboard here, we're showing the universe of splice modulating genetic variants. So each point in this left curve, which is this in this 2D space, corresponds to a splicing mutation. And the sizes of each point denotes the number of people in the world that have this mutation. So smaller dots are rarer mutations and larger ones are more common in the population. And in the top sort of bar, we can filter these mutations based on several categories: which gene contains this mutation, what is the actual variant, what is the workflow which predicted the impact of this variant, and several SBOs, which we can look at the impact of the SBO on the actual exon, on the splicing tracks, and see which ones has the desired effect. And what is the name of the disorder, if there's one associated with this mutation already, that is caused by this mutation, or which is known to be associated with this mutation. So yeah, I would like to emphasize that each one of these points represents a drug program. So there are people in the world who have this mutation, and this is the output of our target discovery system. So we have some confidence to believe that most of these mutations do affect splicing in a way that would cause a disease. So let's look at a few specific examples, beginning with the ATP7B example itself. So if we type out ATP7B, and this is the MET645 mutation, which causes Wilson's disease. In the bar on the right, we're showing some facts associated with this variant. What is the actual sequence? The gene is ATP7B, as we talked about before. There's about 1 to 5,000 people in the world affected by it. These numbers are estimates, of course. And this was predicted by our exon skipping workflow. And this variant is in the deep exonic region. So it's embedded well inside the exon. So that's why it says deep exonic region. And the tracks here are predictions from the splicing model that we discussed. So the purple track is the wild type. So this is the reference sequence. So this is the prediction in healthy individuals. This location in the middle corresponds to exon 6. So in healthy individuals, exon 6 is included in the transcript with around 50%. So 50%. So this is a weak exon. So not all uh, transcripts contain exon 6. And so that's why we don't see a very strong prediction by the model. It does indeed say that the wild type is stronger than the mutant. So the wild type inclusion rate of this exon is around 50%. And the red curve that shows what the mutant looks like. So the model correctly predicts that uh, this mutation causes the exon to look more like a skipped exon, so the exon strength is reduced. And if we introduce some SBOs, let's see what the impact looks like. So we have the first compound, look at compound 2, it has a different effect. But if we look at compound 3, these are some of the compounds that are predicted by our system to work. We see a restoration in the exon splicing, in the exon strength. So the model predicts that if you include this compound and look at the splicing prediction tracks, then the exon strength is restored to wild type like levels. So you would have restoration of exon 6 inclusion. Let's look at a few other examples. So this is just one point in the universe. If we look at another gene called ABCA4, let's look at another type of mutation known as a pseudo exon activation event. So all right, yeah, let's look at this one. So in this case, you have a sequence inside the intron. So again, I'll only enable the wild type and mutant tracks for now. So if you this is a portion inside the intron. So like in healthy individuals, this entire portion is spliced out. So it's all intronic. But in people with this mutation, this specific mutation, you have a portion inside the intron which gets included as an exon. So this is a pseudo exon. This is not meant to be included, but a mutation makes this portion look exon-like. Thus, it's called a pseudo exon. So this, by the inclusion of this portion, you get a non-functional protein. If you targeted it with ASOs, we see that many of these compounds do indeed cause skipping of this mutant transcript. So what we want is this green curve should match the blue curve, and we see that happens with many of the compounds that we've discovered. Another type of variant is called a skip LOF. So if you have a truncating variants inside the exon, you can try to skip that exon itself as long as the exon is non-critical, so that by skipping that exon, you lose those truncating variants and thus restore functional effect. So that's called a skip LOF exon. And in those cases, the desired outcome is to skip. So we want the green curve to be close to 0. And even though I'm only showing a few of the examples selected by our system, there are cases where, for example, this one, where like the model predicts a significant decrease in strength for the mutant plus ASO. So if we go back to the entire universe again, we see that all of these points that we talked about, they're actually clustered by gene. They correspond to people with these mutations. So even if we have a N of one mutation, for example, if we come up with a patient which has a very specific bespoke mutation, which we haven't seen before because our system only takes into account the sequence, it's completely causal, we can predict exactly what the functional impact of that mutation in that person would be and come up with a list of compounds to quickly test which we think would remediate the effect. So even if we have uh, cases of like extremely personalized medicine, we can quickly come up with a list of compounds that remediate the effect of that specific mutation. So in summary, that is a brief overview of our platform. Uh, and as I said, splicing is only one of the mechanisms that we go after. We've developed several other predictors. We have about 30 predictors in our suite of predictors in our AI workbench, and we model things like expression, transcription rate, translation efficiency, ribosome binding, microRNA binding, and so on. But even if we focus on one mechanism, there's so many lives that we can impact by using this technology. And I think that's the coolest thing about working at DG. So thank you for listening and hope to answer your questions during the demo.
All right. Hello, everyone. I'm Shresh again. So I'm just going to address some of the questions that were posted on chat first. Feel free to join in and ask any other questions that you have. So Sangwoo has asked, as far as I know, if one gene is mutated, then the protein will be different than before. Is there any harmful side effect with the new protein? So to this question, so there's many classes of mutations. Not all of them actually change the protein. So if we, for the example that we looked at, you can have a mutation in the intron that causes an exon to be skipped. So if you then restore the exon with an SBO, then you have the exactly same protein as before. In other cases, you have synonymous mutations. If you have an exonic mutation, which is you change the actual sequence, but because there's multiple triplets of sequences that code for the same protein, you can have the same protein even when you change the sequence. So yeah, not all mutations modify the protein sequence, and in many cases, when you restore the splicing impact, you can have the exactly same protein. Or in the case of the Wilson's disease mutation, Wilson's disease variant, even if you have a missense mutation, the change in the amino acid sequence uh, does not always cause an impact. So often the impact is caused by the splicing change. So there's another question from Shirley. How many mutations each genome usually has? Have you looked at the interactions between different mutations if they coexist? Right, so I think having pathogenic mutations is quite rare. So it would be quite rare for one person to have multiple mutations that are pathogenic, although that is possible. And like technically in our system, you can feed it multiple, because it's based only on the sequence, we can model the impact of, similar to how we model like mutations plus the impact of the SPO, you can model the impact of multiple mutations as well.
Yeah, and I'll just say that sometimes we have to do that if there are, very common non-pathogenic mutations that are in the region where we're designing an oligo. We want to make sure computationally that the oligo will have the effect no matter what the genotype of the individuals is. And so that's really, one of the key advantages of doing all this work in silico is that we don't have to actually edit all the cell lines or find a bunch of patient cell lines with all these different mutation combinations. We can just use in silico design to ensure that our oligo will be similarly effective across the population.
Yeah. So next question, I think Yuri has asked, can you clarify the inputs to the neural network model for the exon intron prediction step? How do you deal with unknown splicing mechanisms? So the input to the neural network model is just the sequence of the pre-mRNA. So like you encode the sequence as a 4-channel input. So you convert each position as, like if it's A, then the first channel is 1. If it's C, then the second channel is 1. G, then the third one. T, and then the fourth one, and everything else is 0. And yeah, you just feed in the sequence, and you predict whether each position is either exonic, intronic, acceptor site, or donor site. All right. I see. So Shirley has asked, for transfer learning, what do you transfer? Weights, features? So you do transfer the features. So like you take the features from the neural network model. Like you input the sequence, you take either the intermediate representations or the final outputs, and then you add a few other features. For example, the features that I mentioned, thermodynamic properties, the location of the ASO, distance, etc. And then you use those features as input to another model.
I see there's quite a few new attendees on the right, and so if people have questions, feel free to use the Q&A feature to post them, and we'll answer them.
Mm-hmm. So Seon has asked, for predicting SBO effect or exon detection, have you developed any uncertainty estimation techniques to filter SBOs or targets? Yeah, that's a good question. So for our SBO predictor, we've used a combination of techniques. Because we have multiple genes in which we've screened the ASOs, we've created an ensemble of predictors, and we usually use both the mean prediction of multiple predictors in the ensemble and the standard deviation. We've also tried approaches which have uncertainty estimation built into them, like Mondrian forest, and use that as well. So yeah, I mean, essentially you can imagine if you're trying to prioritize which SBOs to screen, you have to build an acquisition function, which is what is the predicted effect and what is the uncertainty of this prediction. And you can imagine using a combination of both of those things. All right.
Trish, I don't know if you saw Shirley has another question.
Okay.
It's in the chat, not the Q&A.
Okay. So Shirley has asked, I'm also wondering how robust are your methods? For example, do they depend on the data or how you split the data to train and test? What kind of data can lead to better inference? Yeah, that's a good question. So we do make sure to, we do try to make sure that we don't like overfit and make sure that we have enough validation metrics for our models. So if you remember for the splicing predictor, we showed that we had a performance on the test set of the model itself. So on the chromosomes that it's not trained on, we look at the splice site prediction performance on that. But in addition, we also had several mutation databases, which are, you can imagine them as like orthogonal tasks, which we use to benchmark how well our model generalizes. So it's for these sort of models, which are actually used to guide experiments and make scientific discoveries, it's very important to make sure that the model generalizes to something useful. And yeah, so it is necessary to have orthogonal validation data sets. So that's for the mutation predictions. For the SBO prediction, that is true as well. So like you can imagine, different genes would have separate properties, and like if you have the train and test set on data from the same exon, then you sort of overestimate the performance of a model. So it's important to make sure that your train and test set are drawn from the same distribution, but they don't overlap in a very trivial way. And making sure that they're well separated is very important to estimate the generalization performance.
There's another question from someone who's anonymous saying, with the compounds, how do you implement them in the model?
Where is this question?
It's in the Q&A.
Okay. Yes. So for the compounds, we've experimented with multiple different ways. So one thing is, like, you can imagine taking the location where the ASO is intended to hybridize to. So the location in the actual target pre-mRNA where the ASO is sort of designed to be complementary to, and masking out that sequence. So imagine I mentioned that the input is a one-hot encoded sequence. You can mask out, you can set it to either 0 or you can set it to 0.25. Essentially set a portion of your input where you intend the ASO to hybridize to. You can set that to a masked value and see how the predictions change. So essentially it's a way of just estimating how much importance does the model place on one specific location for the final predictions. So how important is this given location for this exon to look exonic, or like how important is it for the exon strength? There's multiple ways of doing it. You can imagine taking the gradients and multiplying it with the inputs. In practice, we found that just simply masking the input works well. You can also replace it with a random sequence and so on. And we've experimented with that as well. So essentially we take the part of the input where the ASO is supposed to hybridize to. We take into account the number of mismatches, have that as another feature, and just take those predictive features from the model. I think, I see another question from Marco. Do you use one-hot encoding of your sequence on input? Yes. Prediction, according to the ROC curve, looks good. However, how many instances did you use to train your model? How many true positive and true negatives? Have you looked at the precision recall plot? Yeah. Have you tested your compounds? As far as I know, there are about 500 mutations known to be associated with Wilson's disease. Do you also consider these mutations? So I'll try to answer them one by one. Predictions according to the ROC curve looks good. However, many instances did you use to train your model? So we don't actually use any mutation data to train the model because the model only looks at the sequence. If you're asking about the first plot that I showed for the test set performance, the model is trained on, I believe, about 1 million examples, 1 million base pairs. No, sorry, 1 million examples of 5,000 base pairs each. The portion that I showed on the plot, those were on chromosomes which the model had never seen. How many true positive and true negatives? So you have one in, like, the number of positives is around 1%. So the class split would be 99 to 1, roughly. So the true negatives, it's for splice sites, it's very hard to say what a true negative is. You can imagine, like, the positives are things that have shown up in RNA-seq experiments, but there's always the possibility of not observing a potential positive. So you have more confidence in the positives than you do in locations being negatives. But yeah, we have looked at the precision recall plot as well. And like the ordering of the predictors is consistent with what we showed in the ROC plot.
Sure. Do you mind if I take these the last two parts of this? Sure. So the question is, how do we actually test the compounds? So Deep Genomics isn't just a computational company. We have about 30 experimental biologists who work for us, and we have 2 lab facilities in Toronto that do cell culture work. And so all the oligo data that Suresh showed comes from cell culture transfections that we did in our own lab, and then measured using either qPCR or a quantitative RT-PCR. And then the fourth question is, there are about 500 known mutations that are pathogenic that cause Wilson's disease. Do we consider those mutations? So the DG12P1 is specifically designed to treat the splicing defect caused by the MET645 ARG mutation. And then we treat, the other, Wilson disease is a recessive disease, so people have mutations on both alleles. And then whatever mutation is on the other allele, we really don't worry about. We think that by restoring that exon 6 inclusion, the productive transcript from that one allele will be high enough to treat the disease. And then the second thing I'll say is that we are planning or considering, developing a drug in a similar modality that will treat more patients using a different mechanism.
Amit, there's another question which I think you're more suited to answer.
Sure.
The
Yeah. So, okay. Are we going to answer Shirley's question as well?
Oh, sorry, I was looking at the Q&A button. Yeah.
Okay. This is the order is confused. Okay. Okay. I'll answer the side effects one first. Okay. Can you predict the side effects of compounds on the other parts, different part of the sequence? And so the answer is yes. What the model is given is the input, the mRNA sequence and then the sequence of the oligo. And from that, it predicts whether or not it'll cause a change in splicing. And so it doesn't matter if that's the region you're trying to affect splicing or just a region that happens to have a similar sequence to the region you're targeting. And we were able to do a pretty good job of using machine learning to make those predictions. And we actually published a paper on bioRxiv earlier this year that looks at off targets caused by steric blocking oligos and our ability to predict them. And so you can take a look at that if you're interested in off targets.
All right. So another question from Shirley. You showed your skipping model has a correlation of 0.6. Is that sufficient for predictions? What is the successful rate when you test the predictions in the lab? So I believe the correlation was actually 0.8. Yeah. And it is sufficient. Like, yeah, so it is very accurate. Like for most cases where it's possible to, so we've like benchmarked our approach to random tiling and using other predictors. And what we found consistently is that our predictors allow us to screen about 10x fewer compounds. So if you rank the compounds using our predictors versus if you use random tiling or use simple predictors, then the rank order for ours is about 10x better. So there's another question by Danish. Are most of the diseases affected by exon splicing orphan diseases? Also, what other diseases have Deep Genomics identified for SBO treatments? Amit, do you want to get this one?
Sure, yeah. So for the universe of targets that Suresh was showing in the demo, those are all Mendelian disease. And I'm pretty sure most Mendelian diseases are orphan diseases. And so they're rare or ultra rare. There are some splicing isoform changes that cause disease in more common or genetically complex diseases. Like I believe there's one in Parkinson's, but you're right in that most cases where exon skipping is applicable, it's a Mendelian rare disease. And for other targets, we have a growing pipeline, but none that we're ready to publicly talk about yet, unfortunately. So there's a question from Yuri. What effects, if any, did intron sequence repeats have on the model? And do we account for them during training?
So we don't really account for them any differently. Like we just make sure to, we just sort of do like chromosome-based cross-validation and test using an orthogonal data sets. But yeah, that's something we can look into. But my feeling is that because if the question about the GC distribution being different for different exons, because we model such a large amount of sequence context for each prediction, that should be, and because the models are, like it's about 100 layers deep, so it should be able to account for that. Right. So another question from Shirley: can you predict drugs that are significantly better than your training set? Do you have to use GANs to be able to predict much better drugs? So I think the space of drugs that we design is just a space of 20 like characters made up of ACGT of length about 18 to 22. And so the space of design is like clearly specified. You can enumerate all of them, but it's very large. And you don't need to generate new kinds of drugs because they're like fully specified as far as the sequence content. Like you can play with the chemistry and all of that, but you could, as far as the sequence content is concerned, you can enumerate all of them. And we don't. So for the examples in the scatter plot that I showed, none of like that was on a held out set. So all of the compounds that we were making predictions on, those were not in the training set. So all of the predictions we made were on compounds not included in the training set. I hope that, and yeah, I mean, I'm sure.
I just have, well, I have two things I just want to add to the answer, Suresh. One is that there's a drug of this class that is approved. It's called nusinersen or Spinraza. And one of the first things we did when we were building oligo predictors is we ran the approved drug through our model, and it confirmed that the model thinks that drug should have the right effect. But then we also identified oligo in the same region but not identical that we predicted to have an even stronger effect than this approved clinical drug. And when we tested that in our lab, the drug that our model thought would be higher would have a stronger effect did in fact have a stronger effect. And I just want to clarify one thing. So 4 to the 20 is kind of big, but the reason why we can easily enumerate all drug candidates is that we're only interested in ones that are perfectly complementary. They're perfectly complementary to the pre-mRNA, and so that for a given exon, there's maybe thousands of compounds or tens of thousands at most, and so you don't need to use a GAN or other generative approaches to find the best oligo in that set. So I think our time is pretty much up. And so, yeah, I just want to thank everyone who is on this call for attending. And yeah, if people have more questions or interested in the work that we're doing, don't hesitate to reach out to Suresh or myself. We're happy to talk science.
Thank you, everyone.